So next speaker is Mahekta Gujar from Eric Lundquist lab in Lawrence University of Kansas.
Right. First off, I would like to thank the organizing committee for giving me this opportunity to present my research work in front of all you wonderful people. So the Lundquist lab is interested in understanding how guidance molecules and their receptors affect growth cone dynamics during growth cone outgrowth. Several guidance molecules and their receptor functions have been extensively studied, however, post-development. So UNC-6 Netrin is a secreted guidance molecule that serves as a bifunctional signal, that is, it attracts certain axons, whereas repels others during development. And our lab has found a correlation between attraction and stimulation of growth cone protrusion, as well as repulsion and inhibition of growth cone protrusion. In an UNC-6 repelled VD growth cone expressing both the receptors, UNC-40 and UNC-5, loss of the repulsive cue UNC-5 leads to a larger growth cone and more protrusive growth cone as compared to wild type. To look at Netrin-mediated stimulation versus inhibition of growth cone protrusion, we use several tools, including the loss-of-function mutants of the receptors UNC-40, which mediates attraction, and UNC-5, which mediates repulsion. We also use the constitutively activated versions of the receptors by adding an N-terminal myristoylation signal to the cytoplasmic domain, enabling them to be constitutively active even in the absence of UNC-6. So as I mentioned before, we have seen a correlation between attraction and stimulation of growth cone protrusion. In UNC-6 Netrin-mediated attraction with UNC-40, we have seen that UNC-40 stimulates protrusion through a pathway that involves CDC-42, the Rac GEF TIM1, as well as the Rac GTPases, SET10, and MYR2. And it is interesting to note that several of these molecules affect both attraction as well as repulsion. On the other hand, UNC-5 is a repulsive cue, and we've seen that it inhibits protrusion of growth cones through a pathway involving the Rac GEF UNC-73 trio, the Rac GTPases SET10 and MYR2, as well as the potential microtubule interacting protein UNC-33 crimp. And I will talk more about these molecules in this pathway and their effect on growth cone cytoskeleton, as well as microtubule distribution. So as I mentioned before, a repulsive response is necessary to limit growth cone protrusion, and this can be seen here in these UNC-5 mutants. Loss of UNC-5 leads to this overprotrusive growth cone phenotype with larger growth cone area as well as longer and longer lasting philopodia as compared to wild type, suggesting that UNC-5 does play a role in inhibition of growth cone protrusion. On the flip side, when we look at these activated versions of UNC-40 and UNC-5, we see that the repelled VD growth cone is inhibited, that it is much smaller as well as less protrusive with hardly any filopodial protrusions coming out. And this suggests that UNC-40 as well as UNC-5 both play a role in inhibition of growth cone protrusion. Furthermore, our lab has also shown that a functional UNC-40 is necessary for this overprotrusive growth cone phenotype observed in UNC-5 single mutants. As you can see here, UNC-40 and UNC-5 double mutants do not exhibit this overprotrusive growth cone phenotype as seen in UNC-5 single mutants alone. And thus we speculate that Netrin and its receptors maintain a balance between protrusion and retraction within the same growth cone, which results in proper guidance. And today I'm going to talk about a potential mechanism by which UNC-5 mediates inhibition of growth cone protrusion, and this involves the microtubule cytoskeleton. So the growth cone is a highly dynamic structure which is composed of microtubules as well as actin. Microtubules are generally present in the central domain, continuous with the axon shaft, whereas actin filaments and bundles are present at the periphery of the growth cone. It is known that a myosin-dependent retrograde flow of actin pushes the microtubules and compacts them into the central domain. And this suggests that microtubules are extremely rare in the growth cone periphery. Our lab has looked at localization of F-actin in these VD-repelled growth cones, and what we see is that in wild-type VD growth cones, F-actin preferentially localizes to the dorsal or leading edge of the growth cone. However, in Netrin signaling mutants, such as UNC-5 and UNC-6, we see that this actin localization is randomized throughout the growth cone, or sometimes showing a more ventral bias. So I've told you that Netrin signaling affects polarization of F-actin. I've also mentioned that UNC-33 CRMP plays a role in inhibition of growth cone protrusion along with UNC-5. So UNC-33 is a microtubule interacting protein, and another molecule, UNC-44 anchoring, localizes UNC-33 to the axons. And we have shown that UNC-33 and UNC-44 mutants display this increased large growth cone phenotype very similar to UNC-5 mutants, with longer and longer lasting filopodia as compared to wild type, suggesting that UNC-33 and UNC-44 both play a role in inhibition of growth cone protrusion. UNC-33 is a potential microtubule interacting protein, and we wanted to assay the role of UNC-33 in microtubule distribution in the VD growth cone. And to do this, we use the microtubule end-binding protein, EBP-2 tagged with GFP, and it's also been shown that EBP-2 labels the microtubule plus ends in vivo in C. elegans. When we look at the expression of EBP-2 GFP in the VD axons and growth cones, we see EBP-2 puncta nicely localizing all along the axon, as well as in the proximal region of the growth cone, with very few or no microtubules present, no puncta present in the growth cone periphery. And this suggests that EBP-2 puncta are rare in the growth cone periphery. When we quantify the amount of EBP-2 puncta in wild-type VD growth cones, like I mentioned before, there are very few EBP-2 puncta present in the growth cone periphery. However, when we look at several UNC-33 and UNC-44 mutants, we see a significant increase in the number of EBP-2 puncta in the growth cone as well as the filopodial protrusions. And this suggests that UNC-33 may prevent microtubules from entering the growth cone periphery. As I mentioned before, UNC-33 inhibits growth cone protrusion mediated by UNC-5 or along with UNC-5. And so we wanted to look at these UNC-5 mutants. And what we saw was very interesting. We see an increase in the number of EBP-2 puncta in the growth cone periphery as well as in the filopodial protrusions, suggesting that the Netrin signaling receptor UNC-5 mediates inhibition of growth cone protrusion by preventing microtubules from entering the growth cone periphery. On the other hand, when we look at the activated versions of UNC-40 and UNC-5, which show those inhibited growth cones, we see a significant decrease in the number of EBP-2 puncta in the growth cone periphery. And this data suggests that EBP-2 accumulation in the growth cones may be a more pro-protrusive effect rather than an anti-protrusive effect. So our lab has shown previously that UNC-73 trio inhibits growth cone protrusion along with UNC-33 and UNC-5. And UNC-73 mutants show these large growth cone, over-protrusive growth cones very similar to UNC-5 and UNC-33. And to assay the role of UNC-73 in microtubule distribution in the VD growth cone, we use two alleles, RH40, which completely knocks out the function of the Rac GEF domain of UNC-73, as well as E936, which affects several isoforms of UNC-73. And what we saw was pretty interesting because, as you can see here, in the UNC-73 RH40 mutant, we saw extremely few EBP-2 puncta in the growth cone periphery and in the filopodia protrusions. However, E936 showed a significant increase in the number of EBP-2 puncta as compared to RH40 and wild type. And this suggests that microtubule accumulation in the growth cone is not a secondary consequence to an enlarged growth cone phenotype. And so we speculate that one way the inhibition of growth cone protrusion is mediated by UNC5 is maybe by excluding microtubule plus ends from the growth cone periphery along with UNC-33. And furthermore, our UNC-73 experiment and result tells us there may be a split in this pathway, in this inhibitory pathway, one involving UNC-33, which prevents microtubules from entering the growth cone periphery, and the other involving the Rac GEF, UNC-73, which controls F-actin polarization, both mediating a balance in growth cone protrusion. And so in conclusion, we've seen that Netrin and its receptors regulate growth cone protrusion that correlates with attraction versus repulsion. Furthermore, Netrin and its receptors maintain a balance between protrusion and retraction within the same growth cone. We've also delineated a new pathway involving UNC-33, the Rac GEF, UNC-73 trio, and the Rac GTPases that mediate growth cone retraction in response to UNC5. And also, UNC5 might restrict microtubule plus ends from entering the periphery of the growth cones to prevent protrusion. And with this, I would like to thank all my lab members, and especially my mentor, Dr. Eric Lundquist. And also, I would like to give a quick shout out to Dr. Adam Norris for his amazing contribution for this axon guidance project. Thank you.
So the effect on microtubules, you think that it's separate from the effect on actin, but it's been shown in cultured cells that actin retrograde flow actually doesn't allow microtubules to move into the lamellipodia.
Right.
So could it be that the effects you see on microtubules are a indirect effect of the changes in actin?
Yes, that's why we see in the Netrin polarization, in this Netrin signaling mutants, that actin is not localized properly, and that's why probably the microtubules come out. And I've also seen the actin localization in all these mutants, and they are mislocalized. So it could be both.
So do you think that motor proteins might be involved in also this
I'm sorry?
motor proteins such as kinesin?
It's possible, yes, they could be involved.
Because UNC-116 has been involved also in this process. So did you specifically test that?
No, I haven't yet tested that.
This is a really naive and general question, but when a growth cone retracts, like you showed, what do you envision is actually happening? Is the membrane being endocytosed, or what's happening? How does retraction actually happen?
So in the inhibition of growth cone protrusion with the large growth cone phenotype, so I think it's more of just, we know that actin is mislocalized in that. I'm not too sure about the endocytosis part of it, but I think it's just because it's missing that repulsive cue, so it's not maintaining that balance. And that's why we see that over-protrusive growth cone phenotype. So, does that answer your question?
Okay, so if there are no further questions, we thank.
Thank you.
Thank you.